Gene therapy has emerged as a promising approach for treating a wide array of genetic disorders and diseases. One of the innovative delivery methods in gene therapy is the intra-cerebroventricular, or ICV route, which involves administering therapeutic agents directly into the CSF within the brain's ventricles. My name is Julie Fourgette, and I will be presenting on ICV administration in mice with respect to potential effects from the selected pain management and the injection procedure on animals' behavior post-administration. Functional Observational Battery Assessment, or FOBs, were performed at 4 and 24-hour post-dose and included assessment of the behavior, sensory-motor functions, and physiological responses. This study was designed with two groups: the positive control group that was administered buprenorphine ER and meloxicam without undergoing the surgical procedures, and the surgical group that underwent the ICV injection with saline solution for injection. This second group also received the same pain management as the positive control group. Animals in both groups displayed a more active behavior compared to the pre-dose assessment, indicating a positive effect of the selected analgesic on the general behavior. For animals receiving the ICV injection and the analgesic drugs, activity levels was comparable between the 4 and the 24-hour post-dose time point. However, unusual posture such as strap tail and hunch, as well as abnormal gait, mainly tiptoeing, were noted at a higher incidence and greater severity for the ICV group compared to the positive group. This may be an indication of a possible exacerbation of the condition induced by the ICV injection. Further refinement for the analgesic selection may be necessary to mitigate the increased activity observed at 4-hour post-dose, to improve the ability to differentiate potential TA-related effects on the behavior at this time point. Join me on that poster for a more in-depth conversation on the topic, and hope to see you in great numbers.